How to distinguish muscle tissue types under a microscope. Have you ever looked at a microscope slide and wondered how to tell different muscle tissues apart? Understanding muscle tissue types is essential in the study of human anatomy. Let's break down the three main types of muscle tissue: skeletal, cardiac, and smooth muscle. Each type has unique features you can identify under a microscope. First up is skeletal muscle. When you look at skeletal muscle fibers, you will see long, cylindrical shapes. These fibers are multinucleated, meaning they have multiple nuclei located at the edges of the cells. One of the most noticeable features is the striations, which are alternating dark and light bands. These bands are due to the organized arrangement of proteins called actin and myosin. The fibers are arranged in parallel bundles, making them easy to distinguish. Skeletal muscle is responsible for voluntary movements, like moving your arms and legs. Next, let's talk about cardiac muscle. This type of muscle is found only in the heart. Under the microscope, cardiac muscle fibers appear shorter and branched. They usually have one or two nuclei located in the center of the cells. Like skeletal muscle, cardiac muscle is also striated, but the striations may not be as clear. A unique feature of cardiac muscle is the presence of intercalated discs. These dark lines appear perpendicular to the fiber direction and help the heart muscle cells contract in sync. Cardiac muscle works involuntarily to pump blood throughout the body. Finally, we have smooth muscle. This type of muscle is found in various organs, such as blood vessels and the digestive tract. Smooth muscle cells have a spindle shape, which means they are elongated with tapered ends. Each cell has a single nucleus located in the center. And like skeletal and cardiac muscle, smooth muscle does not have striations. The contractile proteins in smooth muscle are not arranged in the same organized way, giving it a more uniform appearance. Smooth muscle controls involuntary movements, helping organs function without conscious effort. Now, how can you identify these muscle types under a microscope? Start by using staining techniques like hematoxylin and eosin. This will help you see the differences more clearly. Skeletal and cardiac muscle will stain pink and show visible striations, while smooth muscle will appear more uniform without any striations. Pay attention to the position of the nuclei. They are peripheral in skeletal muscle and central in cardiac and smooth muscle. Look for intercalated discs to confirm you are looking at cardiac muscle. Also, remember the shapes of the cells: cylindrical for skeletal muscle, branched for cardiac muscle, and spindle-shaped for smooth muscle. Lastly, consider the context of the tissue. Skeletal muscle is attached to bones, cardiac muscle is found in the heart, and smooth muscle lines hollow organs. By recognizing these features, you can accurately distinguish between the three types of muscle tissue under a microscope. Understanding these differences is key to grasping how the muscular system supports and controls movement in the human body.